Hi, everyone. Welcome to NDF's GNEM Symposium Speaker Series. We're so glad to have you here joining us for another week's presentation. Today, we're really excited to have with us Dr. Noah Weisleder from The Ohio State University. He's been a friend of NDF's for a couple of years now and has been able to present to us at our live symposiums, and we're so glad that he was able to pivot along with us to this online forum. He's going to be speaking today on targeting membrane repair in GNE myopathy. So without further ado, I will leave it to you, Noah. Thanks so much for joining us.
Thank you very much, Nancy. I appreciate the opportunity to present today. I'm always excited to present both to the scientific community, but more importantly to the patient community that's interested in GNE myopathy. That's the really one of the reasons I got involved in the field of biomedical science as opposed to working in a lot of other fields, was the opportunity to work with the patient populations and pursue treatments and therapeutics that can actually have a deep impact on the lives of patients who suffer from neuromuscular diseases. So it's an exciting opportunity to be here today, both to talk to the scientific community associated with looking for treatments for GNE myopathy, but also to talk to the patients as well. And I've recently come to this field. I mean, traditionally, my laboratory has been much more interested in other types of neuromuscular diseases. We've worked in limb-girdle muscular dystrophy and Duchenne muscular dystrophy in the past, but it's a great opportunity today to show some of the initial work that we're doing looking at GNE myopathy. My laboratory is currently at The Ohio State University. I am a professor and director of graduate studies in the Department of Physiology and Cell Biology there. I also head up the postdoctoral office at the university level at Ohio State. I started my laboratory at Rutgers University before moving here to Ohio State a few years ago. But I also did a postdoctoral fellowship at the University of Medicine and Dentistry of New Jersey, and then also did my graduate work at Baylor College of Medicine in Houston, Texas. So today I'm excited to talk to you a little bit about some of the work that we've done looking at GNE myopathy in my laboratory. And I'm going to show you some things that build upon the direction that we have when we move towards looking at GNE myopathy. And a lot of that is recent work we've done in our laboratory relating to a process called membrane repair. And I'll tell you a little bit more about how that works and how it can contribute to different types of muscle diseases like the limb-girdle and Duchenne muscular dystrophy I talked about earlier. And then I'll talk to you a little bit about what we do to develop therapies that target membrane repair and how those might be applied to the treatment of GNE myopathy based on some of the preliminary results that we've gotten that led to funding from the NDF to pursue in our laboratory. Before I start, I do want to make a few disclosures. When I was on the faculty at Rutgers, they spun off a biotech company called Medicine, and that's developing one of the things I'll talk about today, a recombinant human MG53 as a therapeutic approach. So it's important you realize that that's something that I am both the founder in and I'm currently a shareholder in. I also receive research funding and I'm a shareholder in Myophinity as well. They're also interested in different types of membrane repair proteins. And then finally, I also receive research funding and consulting income from Casma Therapeutics, who also work on therapeutics for different types of neuromuscular disease, but not relevant to what we'll be talking about today directly. So today, I'll talk to you a little bit about what we study in my laboratory, and one of the processes that we study is membrane repair. I'll show you how membrane repair works and how we can target these membrane repair mechanisms as a treatment for different types of muscle disease. And then I'll tell you some of, like I said, the work we're doing in collaboration with the NDF is looking to see if there's defects in membrane repair in GNE myopathy, and if this is something that we can target as a therapeutic approach in this disease state. So I always like to start with this very fundamental idea about what cell membrane repair is. So we all know what a cell is, right? That's the fundamental building block of everybody's body. The tissues that make up your body are built up out of individual cells. And what makes an individual cell, what really defines what a cell is the membrane that surrounds it. And this membrane is very important because it allows the cell to do a lot of different processes that would normally do. And it's essential that this membrane remains intact, because if you break down this membrane, you really don't have a cell anymore. And that cell will die through a process that we call necrosis. And you can see that this cell breaking apart over here. But the cell has developed a lot of types of processes that allow it to repair disruptions and allow it to survive a break in the membrane that would normally cause that cell to die. And this is what the cell is doing over here after it repairs. So When we look at the sort of another representation of the cell here, there's that break in the membrane, and the influx of components into the cell triggers the formation of a repair patch. And you can think of this as a lot like the patch you might put on a tire, right? If you don't patch this disruption quickly, then you'll have the intracellular components flooding out and the intracellular components or the extracellular components flooding in, and you lose your cell, which is what you see over here. But this patch that it forms is what will protect the cell from death and allows you to maintain more cells that would normally survive some sort of insult to a tissue. And this process occurs in a lot of different types of cells. You can see just from the list here, you can see that in a lot of different parts of the body, the cells in that part of your body go through a repair process similar to what I just talked to you about. And this kind of shows you how important it is to be able to have that repair process. But where it's most important is when we talk about skeletal muscle and cardiac muscle. And that's something that makes a lot of sense, because when you have a cell that contracts like muscle does, you're going to put a lot more mechanical stress on that membrane, right? It's being torn a lot more than it normally would if the cell was just sitting there. So you need to have that active repair process in order to replace all those disruptions that take place. And who hasn't, after exercise or that sort of thing, been sore? And that's actually those little tears that occur, or just in the course of normal physiology, you get little tears that occur that have to be patched. And that's why you see this is much more common. 3 to 20% of your muscle fibers in your skeletal muscle will show some signs of repair at any given time. And that's true also when we look at cardiac muscle as well. Both of these contractile tissues require a lot of membrane repair to be supported. And we talked a little earlier about that patch that's formed, the patch that occurs when you have a break in the membrane. And that's exactly what I'm showing you here. But this is electron micrograph. So this is the most powerful microscope we have. And we're using this to visualize a cell here. You can see the cell. And here, this is an uninjured cell. And then we look at all three of these other images. These are where the cell's been injured. And we fix the cell immediately to see the vesicles start to form. And you can see here, right after the injury, you can see vesicles starting to move towards the injury site. And then they form this large frothy patch that's occurring here. And that's represented over here in this illustration, where you can see again a break in the membrane. The green line's the membrane here. And these vesicles here shown by these red circles, they are all going to traffic to that injury site, fuse with each other, and then with the membrane itself to create this repair patch. That's what I showed you earlier in that green illustration, is it's forming a repair patch that's going to allow the cell to survive a break that would normally cause that cell to die. And what we work on in my laboratory is the actual proteins, the genes that regulate this repair process. And my laboratory and many others have found several genes and proteins that are important for regulating this repair response. One of the most common, one of the first to explore was dysferlin. You can see that here. And we work on a gene called TRIM72, or also known as MG53. And these are both important. As you can see, they're on these little vesicles. They're important for getting the vesicles to the injury site. And when they're at the injury site, the dysferlin helps them fuse together and form that repair patch that we were talking about earlier. And since we came up with this more simplified model, there's been more and more genes that have been linked to this disease state. Some of those are listed here, and but you can see how it forms a very complex structure in the membrane that forms this repair patch and allows cells to survive damage that would normally cause that cell to die. So this is another illustration of that, even more genes that come into play here, which you don't really need to know. I don't want to complicate this more than it needs to be for this talk. But it's important to remember that the MG53 and the dysferlin molecules are very important for the repair response. And a lot of work has been done in this, both in my laboratory and in others. You all know one of your scientific directors, Angela Leck. We actually have worked together in the past looking at some of these genes as well. Her doctoral research focused on both dysferlin and MG53 and membrane repair. And you can see that here. This is actually work that she did where these are cultured muscle cells viewed under a microscope. And what you see in red is MG53 and what you see in green is dysferlin. And what she's done here is use different types of small particles to injure the cell. So you see all these little holes that have been created in the cell here. You can see them all spread around, little circles. Those are disruptions in the membrane that were created by what we call a gene gun. And you can see that at those sites, you see both intense red signal, meaning MG53 there, as intense signal from dysferlin as well, which is seen in green. So those two genes are there, they're working together in that repair response. And you can see they're both at all of these repair patches that are forming. That's why you see it in yellow, because they're both there. So that shows you that there is an important function of these two genes in repair, and that they're working together to form these repair patches that allow the cell to survive. But I'm going to focus most of my talk today on the molecule that we work on in my lab, MG53. Which, as I mentioned earlier, also is known as TRIM72. I'll use those interchangeably as I talk along here, but they're both the same thing. They're both referred to the same protein in your body. And that protein is expressed almost exclusively in skeletal muscle and in cardiac muscle in the heart. You can see that here in this illustration. What these two indicate is that all these other tissues don't have any MG53 in them. So there's none in the lung, the kidney, the liver, the brain, but you see it in skeletal and in cardiac muscle. And that goes back to that idea I talked about earlier, right? Where these are contractile tissues where there's a lot of damage. So you need to have a lot of the repair process there for that to take place. And that's one of the reasons you see so much MG53 in those tissues. And there's a lot of data here. I don't need you to go through all of it, but just two things I wanted to show is here's another repair patch that we form on a muscle fiber. You can see the muscle fiber here in the bright field image, the microscopy image. And then we use a special technique to localize where the MG53 is. And you can see it's also forming a repair patch there, in this case in red. And then going back to that green color for MG53 in a much higher magnification, you can see again one of these repair patches forming. So again, MG53 at the repair patch. We know it's there. We know it's important for the repair to take place. And to drive that point home a little more clearly here, I'm actually going to show you one of the movie images that were recorded in the microscopes in our laboratory, where again, the TRIM72 or MG53 is shown here in green. And what I show down here is the time elapsed. So right now we're at -5 seconds before we're going to injure the cell with a laser. We're going to do that right up here. You're going to see it pretty clearly. And then we'll see how quickly the MG53 translocates to that injury site. I'm going to start the movie now. And you can see, even just a few seconds after injury takes place, how quickly that repair patch forms. And it's very stable by 20, 30 seconds after the injury takes place. And this is essential in order to maintain the integrity of the membrane. We have to patch that quickly. And it's the TRIM72, MG53 that is responsible for that patching taking place. And if I show that movie again rolling here, but I'll also show you a couple of still frames from that. You can see how quickly that patch forms. It's stable by 14 seconds, and it remains all the way to the end of that video at essentially that size. So it's a very strong patch that's formed, and that's to help maintain these muscle cells when they're injured, that they can repair themselves effectively and avoid the muscle cell dying. But what happens when this repair process is not active? Well, that's what we see here. So this is a knockout mouse. So you've heard in this series about knockout mice. Those are mice where we manipulate the genome of the mice to basically remove one of the genes and see how that affects some aspect of how the mouse functions. And in this, we're removing the MG53 gene. And what we see taking place here, comparing a wild type mouse, the histology of that muscle, skeletal muscle here, looks very normal. And in a MG53 knockout mouse, at 3 months, there's some changes. You see some centrally located nuclei, but it still looks pretty normal. You don't see a lot of changes there. But as these mice age, they develop a pretty profound myopathy. You can see that now there's a lot of fibrosis in this muscle. There's difference in sizes of the muscle fibers, a lot of central nuclei. All of these are signs that you see in many different neuromuscular diseases, and particularly in GNE myopathy. You see these sort of changes in the muscle structure occur as you move forward in age. So we see something here that's producing a myopathy that's a late onset adult myopathy, where there's a progressive development of pathology as in this case, these mice age. And that's associated with a loss of force production. You can kind of see that here, a wild type mouse in black, and the MG53 knockout is in blue. So you see these bars are much lower, so it's producing less force. And there's also a compromised ability for the membrane to repair itself. Here we're showing that red dye again. You see a lot more of the red dye going into the muscle fibers in the knockout versus the wild type mouse. So that lets us know that there's a breakdown in its ability to repair, and that they continue to show defects in their repair response, and that's leading to this myopathy we see above. And this was some of the basic science we did to understand the membrane repair process in muscle and how important MG53 was for that. But as I mentioned earlier, one of the things that I'm very passionate about and that my whole laboratory pursues is the idea of looking at ways you can not just understand how these neuromuscular diseases work, but also to look at ways to treat them. And one of the ways that we've found we can do that by targeting membrane repair is using the MG53 protein that I just showed you going to those injury sites as a direct therapeutic to try to increase the repair response in skeletal muscle. So that's what we're showing here is one of the ways that we do that. So what you see here is a couple of different images. This is the same movie on both sides. It's just two different ways of representing it. So what you can see over here where you have more of the gray, that is a cultured muscle fiber there. So you can see that outlined in gray here. And what you see up at the top is a micro needle. So there's a very fine needle that we use for a lot of different purposes in the lab. But one of the things we're doing here is we're actually going to poke a hole in the membrane. It's that simple to create a disruption in the membrane. And we're going to do that twice. We're going to do that up here near where the needle is now, and then come over and do another disruption down at the bottom. And that's, so while that gray area is the cell, if you look at outside of the cell, and you can see the shadow of the cell here, you can see a lot of red. And this is that red tagged MG53 I showed you earlier. But in this case, we've cooked that up in bacteria, the sort of bacteria you use to make biotech drugs. This is a lot like what you do with insulin, right? Insulin that's taken by type 2 diabetics. That's generated in a factory where it's grown in bacteria and then processed and purified. We did the same process here with the MG53 protein. We made an additional amount and we purified that and we put it outside the cell, much like you do with insulin, to see if we can increase the membrane repair response that takes place. So without any further ado, let me show you the injury that takes place over here, and then another injury down at the bottom of the cell. And you can see that red labeled recombinant MG53 is locating to the injury site there. Where those two disruptions were, you can see that there's accumulation of MG53. That's a little more clear here in just the protein image. You can see those injuries in the same place. Now you see it's starting to accumulate and also accumulating down here. So in both of these cases, by applying the recombinant protein outside the cell, it's still able to locate that injury site and help to form a repair patch to prevent that disruption from injuring the cell. We look at that with a little more detail here. You can see right here where they're circled, the recombinant protein going to that injury site, locating there and helping to form a repair patch. So this gave us an indication that this is potentially something that we could use as a therapeutic intervention. If we can add in more recombinant MG53 and use that to boost the repair response, is that something we can use to compensate for the membrane repair defects that we see in different types of neuromuscular disease? This is really important because there's a lot of protein therapeutics like this already on the market. I just mentioned insulin. Insulin is it was the first biotech drug. It's generated as a recombinant protein, and it's used to treat type 2 diabetes by injection. And we could envision using this protein by injection to treat different types of neuromuscular disease. So the first disease we targeted with this approach was Duchenne muscular dystrophy. So some of you may be familiar with this. It's a little different than the GNE myopathy we're talking about today. But it's an earlier onset. It tends to affect, and it generally will only affect boys, and it affects them very early in life. There's sometimes diagnosis at 3-4 months of age now, or 3-4 years of age now. And it's very aggressive, degenerative muscle disease where there's a lot of damage that takes place in the skeletal muscle. You can see that occurring here with a lot of type of damage taking place at the structural level. Again, using that red dye that shows a change in permeability called Evans blue dye, because you can see that down here. It's blue in the muscle. You can see a lot of muscle fibers taking up the blue dye, and that blue dye appears red with this particular microscope. So you can see in the muscle fibers, there's a lot of that Evans blue dye appearing. So that shows there's a breakdown in the ability of the membrane to maintain its barrier function. So you get a lot of dye coming into it. So this is a pretty good indication that there's an issue with how well these cells are repairing themselves. So that's how we wanted to target this and see if we boost the repair, could that be protective against some of the symptoms of Duchenne muscular dystrophy. And that's what we did here. Now, we did this in mice. We haven't done this in humans yet, but we're pushing towards that. In this study, we're looking at a lot of the same things we already looked at. We're looking at muscle histology, and we're seeing that in the mouse model of Duchenne muscular dystrophy, the MDX mouse, there's a lot of fibrosis, there's fatty infiltrate, there's central nuclei, there's a lot of things that you see in many neuromuscular diseases, including GNE myopathy. And then when we inject recombinant MG53 over the course of several days into these mice, we actually see less of all of those pathologic hallmarks. There's still some, it's not totally rescued, at least at these doses, but we see a protective response that occurs because of the addition of that. We also see, looking at that Evans blue dye, the indicator of the permeability of the membrane. I showed you that earlier here. We see that in a MDX mouse, yes, one treated with a saline injection, there's very little of the dye, or the dye goes up in a lot of the muscle fibers. When we apply the recombinant MG53, a lot of those don't show up. So we link that increased repair to the increased or the decreased pathology you see taking place in the MDX mouse because of the injection of this recombinant MG53. And that's all quantified here. We don't have to go into that quantification, but suffice it to say, when we look at this mouse model of Duchenne muscular dystrophy, we see protective effects in these mice when we apply the recombinant MG53. And other laboratories have gone on to show this as well. But that's not the only type of neuromuscular disease out there. We're also interested in onset of an adult onset neuromuscular disease, and that's what we're looking at here as one example of that, limb-girdle muscular dystrophy 2B. So this is another adult onset myopathy, where it's, as you might guess from the name, the limb-girdle aspect, it's principally an issue with the muscles of the limb. The respiratory muscles in the heart are pretty much spared there. There's a lot of similarities with what you see taking place in GNE myopathy in this case. But again, it has many of the structural hallmarks that you see taking place in a myopathy. There's a progressive nature. You start off with some damage, and then as the animals age, you get more significant disruption of the structure of the muscle, again seeing fatty infiltrates and fibrosis. And again, looking at that Evans blue dye, another indication that's breakdown in the permeability of the membrane. You can see that here as well. You see a lot more red dye in the dysferlin knockout mouse than you see in the wild type mouse, just like I showed you for the MG53 knockout and for the MDX model of Duchenne muscular dystrophy. They all show a lot more Evans blue dye. And in this case, that's because there's mutations in the dysferlin gene. I mentioned to you earlier about dysferlin. That's one of the proteins that works alongside MG53 in membrane repair. So we were really interested in this approach because this is where we wondered, so if you compromise part of the membrane repair response, can you compensate for that by using more of the protein that's working alongside that? So that's exactly what we did here. And in this study, we used a different type of approach than what I showed you before. In this study, we're using a situation where we isolate out a muscle. You can see that here. I've shown that in gray. We talked about the muscles being isolated out earlier. And then we bathe it in another type of dye. This dye, which we call FM464, that's over here. You can see FM464. But that's shown here by these little circles. When we have this muscle and we injure it with a laser, so we use this laser and this microscope to do imaging, but we can also injure muscle fibers with it. When we do that, you'll see that the dye rushes into the cell, and then once it gets inside the cell, it goes from basically being inactive to being very brightly fluorescent. So that's that red signal we see in all these images over here. And that dye continues to enter into the cell until the membrane repair patch forms. That's the little stars here forming the patch. So when there's a compromised ability for the membrane to repair itself, we get a lot more dye coming in than we would if the membrane repairs itself quickly. So this is an easy way to measure how effectively the membrane can repair itself in a very reproducible way. If we take a look here at an image, some of the images we see here, for example, this we're looking at, in this case, mouse muscles, where this is from the dysferlin knockout, where we're looking to see if there's a compromised repair. And when we do our laser, which is shown here where this arrow is, burns a hole in the membrane, and we have dye enter into this cell after it was injured. You can see the dye right here. It's very brightly fluorescent. So that's how much dye enters into one of these fibers normally, which is a lot more than you would see in a wild type muscle fiber. You'd see something much more like much smaller amount of dye coming in. But when we add in the recombinant MG53 into these dysferlin knockout fibers, so these are a model of this limb-girdle muscular dystrophy, we see that the recombinant protein greatly reduces the amount of dye that comes into these muscle fibers. So again, we're showing that this can protect against the membrane repair defect that we have in these animals, and help prevent some of the pathology that's going to occur in these animals as they age. So this is another model of neuromuscular disease where we can show that there's a repair response that can be increased with the recombinant MG53 and prevent some of this pathology. Now, one of the challenges with this laser injury assay is it's a very convenient way to measure, dynamically measure too. You can image it while it takes place, but we also want to understand what takes place in a more physiological situation, right? Not all that many of us get shot by lasers, but I suppose it happens sometimes, but for the most part, we don't run into that problem. But here's a set of studies where we're actually doing a more physiologic response that takes place. And in this, we've taken those dysferlin knockout mice and we've actually run them on a treadmill. And when we do that, that damages their muscle fibers, just like we talked about earlier. After you exercise, you have that soreness. Those are little tears in the membrane of your muscle fibers that need to be repaired. Because these have a poor repair response, they get a lot more damage during running than a normal mouse would. And that's what you see taking place here. These are 3 different muscles here: the soleus muscle, the extensor digitorum longus muscle, and the gluteus. We tend to look at a lot of different types of muscles so we can be sure we have a consistent finding. When the dysferlin mouse is just sitting there in its cage, it doesn't have all that many muscle fibers that are damaged. But after we run them, you can actually see a lot more muscle fibers show that there's damage. And you can see that over here on these charts as well. The long hashes here show ones that have been run. So in all of them, we see a lot more fibers being damaged when you run them on the treadmill. However, when you inject them with a recombinant protein and then run them on a treadmill, they go basically back to what they were before. So again, the recombinant protein is protecting them against the damage that takes place when they exercise. So we think this is really important because this is a really physiologic response to what takes place in the muscle. They have compromised ability to repair themselves, and that leads to a muscular dystrophy. But if we add the recombinant protein in, we can prevent that damage from taking place, and we can spare them the long-term effects that would take place due to just normal amounts of activity that take place to these animals. So this is what led us to actually working with the Neuromuscular Disease Foundation. So while we've shown in multiple types of neuromuscular disease, and others have shown as well, that the repair mechanisms involved in the membrane are important for preventing neuromuscular disease, it's not in all neuromuscular disease. There's a lot of different things that can cause damage to muscles and cause a loss of the ability of the muscle to repair or the muscle to maintain its function. And thus, you need to understand what's taking place in a particular muscle during a particular disease, so you can begin to target therapeutics towards that. And this is ultimately where we got involved here. We're interested in understanding what the fundamental mechanisms of membrane repair are, but we also want to know how they might contribute to different disease states. And as I talked about, there's several similarities between some of the other diseases we've studied and GNE myopathy. So we were curious if this might be one of the factors that contributes to the progression of GNE myopathy. And as I mentioned earlier, I've worked in the past with Angela Leck, looking at different types of membrane repair processes. So we were naturally interested once they started working on GNE myopathy and started developing the NDF cell bank, which of course comes from the donations of several members of the patient community. And we're very grateful for those opportunities to be able to use those cells to better understand how GNE myopathy works. And why do we need to know how the disease works in order to be able to treat it? Well, while gene therapy is a great approach for putting the gene back in, trying to understand what type of repair defects, or what type of defects contribute to that, whether it is an issue with membrane repair, will help us not just to design therapeutics, but also to design therapeutics that can be used alongside gene therapy. Most of the gene therapies that are on the market, like when we look at Duchenne, or moving towards the market, when we look at things like Duchenne muscular dystrophy, aren't true cures. We know that when we put the gene therapies that are currently being developed into a Duchenne patient, they're actually not going to be brought back to what you would see in a normal healthy volunteer. They're going to still have some compromised membrane function and compromised muscle function. So we would propose that we could use recombinant MG53 alongside those gene therapies as a protective response. But we don't know if that's the case in GNE myopathy, and that's exactly why we're working with the NDF right now to find out if that's the case. So we have a funded project in our laboratory where we're looking at two different things. We're looking at, in the first aim, to determine how much of a repair defect there might be in GNE myopathy cells that come from the patient cell bank that's housed in the LEC laboratory at Yale. And then we also are looking to confirm the membrane integrity defects that we might see in those cells with actual patient biopsies. Like I said before, we're not always going to have just one assay which will tell us what's taking place. We want to see the same thing in multiple assays so that we can have confidence what's taking place is actually important for the disease that's occurring. So the fundamental resource that we're using for a lot of these studies is these GNE myopathy myoblasts that were isolated from patient volunteers. So these were, as I said, generated in the LEC lab. They took skin biopsies from patient volunteers and from also healthy volunteers as well, many cases in the same family, and those provide an excellent opportunity to have great, what we call, controls to understand if what's really taking place is occurring. So the skin biopsies are grown out into fibroblasts, and then through some genetic manipulation, we can convert those into myoblasts. And those are the fundamental building blocks of muscle. They're smaller cells that fuse together to make a much larger cell, which is what becomes the muscle fibers in your muscles. And what we do is use that laser injury approach that I talked about earlier. In this case, this is a little dish of cells here. You can see the myoblast inside, and then we've added in that dye I showed you earlier, that FM464 dye, which is inactive outside the cells. But then when we injure the cell with a laser, or you can see that taking place right here, and then I'm going to magnify this a little bit so that you can see where we're going over here. So magnifying that area, here's your cell being injured, and you can see the dye starting to flood in. And then that repair patch occurring like we talked about earlier. So here we've taken these basic muscle cells, these myoblasts, and injured them and seen how quickly they can repair themselves. And this is some of the initial data that we found here. Here we're looking at 3 different cell lines isolated from 3 different individuals. In that case, there's 2 healthy volunteers and 1 GNE myopathy patient. These are all from the same family, so they have similar genetics. And what we see here is some recordings of how much dye is entering into the cell. But you can really even just appreciate here looking at the individual cells themselves. Here's 2 healthy lines before injury and then after injury. When we injure them with the laser, you see very little dye enter. Same thing is true for this other healthy individual. We see very little dye entry. But when we move over to the GNE myopathy, there's the initial injury that occurs here, and you see that there's a much larger amount of dye that enters into these what we call myotubes than what occurs in just the healthy volunteer myotubes. That's all quantified over here if you want to see that. But the conclusion there is that if we look at a GNE muscle cell, they have less ability to repair their membrane than healthy controls do, which indicates there may be a membrane repair defect there as well. And what we're doing now with the NDF is looking at other cell lines from other patient and healthy volunteers and looking to see if we see similar responses there. And we see the same thing starting to show up in this. This is another set of these isolated lines from the NDF cell bank. In this line, this is another healthy volunteer from the same family as another GNE patient. And again, when we look at that, we see more dye influx occurring in the GNE patient than we have in their healthy volunteers. And again, this seems to be a general trend where we're seeing in multiple GNE patient cell lines, we're seeing a compromised ability of the membrane to repair itself. And this is really important because if this tends to be if this is true, if this is the case, and we see that both in more patient cell lines, as well as in the staining we'll do in the muscle biopsies, then that'll show us that there's a potentially a membrane repair defect that can be targeted through some of the approaches that we and other people have developed to be able to improve membrane repair in neuromuscular disease. So what I'd like to do is just give you a few conclusions here that I've hopefully been able to show you that membrane repair is essential for muscle health. When there's a change in that repair response in Duchenne muscular dystrophy and limb-girdle muscular dystrophy, these lead to muscle pathology, muscle disease that we see appear both in animals and in human patients. And we also show, at least in animals at this point, that we can increase the membrane repair response. And we do that, it can protect those animals against muscular dystrophy. And we hope to be able to move that forward into treating patients in the not too distant future, but that's something that we're working on now. And we use the membrane repair defects. We don't know for sure that these appear in GNE patient myoblasts yet, but the early preliminary data we've done certainly indicates that's the case. And we look forward to working with the NDF to be able to find if that's what's occurring here and whether that's something we can target as a therapeutic approach going forward. There's a picture of my laboratory. Several people in the group contributed to this work. I'd like to particularly emphasize Sayak Bhattacharya, who's actually moved on to Dominion University, where he's doing additional cell membrane research there, now independently. But he did a lot of the sort of laser injury work we see taking place here. Lyubov Gushchina is a protein engineer. She did a lot of the work with developing the MG53 protein. And also Tom Bodnar, who's right here. He's actually he was a PhD student, just graduated from the laboratory. He's also moving off to start his own laboratory group as well. And he's going to continue to do some of this work too. And we couldn't do all of this without working with a lot of great world-class collaborators. One of those is the LEC laboratory. Like I said, we've got these patient cell lines from them, and they've been very useful as well. We've also, as you may know, Nationwide Children's Hospital and Ohio State are associated. Dr. Paul Martin has been a colleague of mine for a number of years, and we've worked together on a few projects. And also Kelly Crow, who's also working with the NDF. We're also working with her to look at some of these patient muscle biopsies and see how different markers change as the disease progresses. So it's been a real pleasure to talk to you today. I look forward to hearing some of the questions and answering those. But first, I want to emphasize some of the key takeaways that you could take with you as you move on and ask additional questions. I want to make sure that you understand the importance of membrane repair for muscle health and how that leads compromised capacity to do that leads to muscular dystrophy. We showed that multiple times, both in human and in animals. And hopefully you were able to see how using different types of approaches like recombinant MG53 to increase membrane repair can help protect against different types of muscular dystrophy. At the current point, we can see that at least in some GNE myoblasts from patients do show a defect in their ability to repair their membrane. We need to test more of these muscle cells to be sure that this is the case, because you can have a lot of variation in human samples, and we need to confirm that there's actually a clear defect there, and we can make sure that that's a reproducible result. And I also want to confirm that in muscle biopsies, where we can see directly in patient muscle that there's a change in the ability of the membrane to resist damage, and that'll help us understand whether we can target membrane repair as a potential therapeutic for GNE myopathy. So thank you all for taking the time today. I really appreciate the opportunity to talk to both the patient community, but also to talk with the scientists who are working the NDF. And it's really been a pleasure to talk with you today, and I look forward to any questions that you might have.
Wow. Thank you, Noah. That was amazing. That was a lot of information, really well presented, so interesting and so promising. I know that people are really interested in hearing more from you as you have more to report. But in the meantime, I do have a couple of questions from people who have tuned in with us. Someone asks from a text, with a damaged GNEM muscle cell, do we then get the patches you are talking about? Are they sealed to stop further damage, and how applicable is the MG53 to them, to the GNEM patients?
Oh, that's a really great question. And that's exactly what we want to do with these muscle biopsies, right? So what we're going to do with those is we're going to take those and we're going to do a little bit of that staining I showed you earlier. I call it IgG staining, but what it really is just showing you is how many muscle fibers were damaged. That's when I was running those mice on a treadmill, right? So we want to do the same sort of thing. Now, obviously, we're not going to take patients and we're not going to run them on a treadmill, but even at baseline, you'll see a lot more disruptions, at least in these mouse models. You'll see, and we've also looked at some human sections and other diseases. You'll see that there's an increase in the number of fibers just at baseline when we compare healthy volunteer biopsies to the biopsies that come from neuromuscular disease. So by doing that, we'll know if there's a change in the functional ability of the membrane to repair. But more directly for the question that's being asked is, do we see those patches occurring? So for that, we're going to do immunostaining for some of these factors. We'll do immunostaining for MG53. We'll do immunostaining for dysferlin. And that goes back to what I was showing you earlier from that, actually some of that work that Angela Leck did as a graduate student. We'll look to see if we see disruptions like that. They'll look a little different because we're not forcing them. But if we see disruptions like that, where there's patches that have formed on the membrane. So that's exactly what we want to do. And we couldn't do that without the support of the NDF to do those sort of studies. And also the other investigators in the field. Like I said, we haven't worked on this before, so we don't have muscle biopsies from patients with GNE myopathy. So it's been very helpful to have both the funding from the NDF to do this work, but also the access to those patient samples, because that allows us to do exactly the experiment which the question is about and figure out how relevant this is for GNE myopathy. And when we know that, we can have a better idea how relevant the recombinant MG53 will be to treat these sort of disruptions that might take place.
Right. Okay. Thank you. Yeah. I mean, we're so proud of our collaboration and our consortium finding people like you and the other scientists who are working together, and whose work can't continue without the help of each other. Really, the biobank that we've been able to build has been great, and I'm so glad to hear that you've been able to take advantage of that. Tara has a question again about the MG53 protein. She said that she might have missed your definition. So she was looking for a definition, and also she was asking how prevalent it might be in foods.
Oh, great. Yeah. Well, I wanted to make this a broadly accessible talk today. So I didn't talk a lot about the specific details of the protein itself, but it's a great question. And
very smart patient.
kind of a inside baseball idea here too about the type of the protein. So this is what we MG53, like I mentioned, also known as TRIM72, is what it's a part of a big class of families. They're called the tripartite motif family of proteins. And these are found in there's a lot of members. The 73, it's the 73rd member of the family. There's over 80 different members of the family. So there's a lot of these type of proteins in the human body. And a lot of them are in different tissues. They're in a lot of different places. And as I mentioned, the TRIM72, the MG53, is expressed almost exclusively in muscle. And that's because it's important in membrane repair. It needs to be there to help boost the capacity of the membrane to repair itself. And specifically because those muscle fibers are contractile and there's a lot more tears that occur in their membrane just in the course of normal function. So we see that a lot in different types of muscle. So it's a very enriched in muscle. It's a tripartite motif protein, and that its aspect in the tripartite motif family, its function in there allows it to be able to participate in membrane repair. And we published a lot of information on how the protein itself works that I won't go into a great deal of detail today, but certainly I'd be happy to share those publications with anyone who would be interested in the specific details of that. But I really enjoy, and I'm interested in that question about whether it's in foods. Now, one of the things, like I talked about, we use this as a therapeutic approach, and we've done this by injection in these animals, and we would envision that it would probably be used as an injection because we've looked at other ways to kind of put it in a oral delivery mechanism, which hasn't worked particularly well. It's kind of a complex protein, so it's difficult to deliver that way. Although there may be other mechanisms we can do to do that with an oral delivery. As you know, with like insulin, which I used as an example before, that used to just be done by injectables and still is in a lot of cases. But there's also oral delivery now for different types of that. So that's also potentially a way that we could deliver the protein. But from the point of view of foods, it is enriched greatly in a lot of, and we talked about it being expressed in muscle. So if you look at different types of meat, there's a lot of MG53 in that. I don't know that that would be helpful as a therapeutic because of the way that it's absorbed into the intestines, through the intestines, you wouldn't get the whole protein through. It'd be smaller parts of that protein, and I don't know that they would be functional. It is interesting, though. There's a group in France that's actually interested in meat quality. So they're an agricultural researcher, and they've looked at the differences in tenderness in meats and what proteins makes meat tender. And one of their interesting findings was that the more tender meat is, the less MG53 it has. So I just think it's an interesting correlation there. We don't have any proof of it yet, but maybe you have less MG53, the muscle cells don't repair as well, and it makes them more tender. It's something we're interested in pursuing, actually, along with them, but something we don't have too much evidence of yet.
That is interesting. Will has asked, and I think you kind of just addressed this, is there treatment in the future for GNEM membrane repair? And if so, would it be an oral medication or an injection like insulin?
Oh, that's a great question. Yeah, and probably that comes from talking a little earlier too about insulin. And I guess I alluded to that, you're right, a little bit about it's as an injection of insulin. Right now, we're thinking about that from the point of view of injection, and with the caveats I talked about earlier for oral delivery. One of the things that we're currently doing in my lab, and one of our corporate partnerships, is actually developing a next generation of the MG53 molecule. It's a smaller version that might be easier to deliver and might last longer in the body. So even if we still had to do injections, we might be able to get away with doing a much longer course or a much shorter course of injection. So right now, much like with insulin, you'd probably be looking at a regular injection, maybe even daily. But we want to kind of spread that out. It's a lot easier to apply if you don't have to inject as often. So a lot of our efforts now are focusing on ways to make that a fully efficacious repair protein, but something that can be used a little more effectively or easily by patients. So we're certainly spending a lot of time looking at ways to make that a better delivery. But right now, what we envision right now is an injection that would be able to be used in different types of neuromuscular disease. Right now, we haven't tested directly whether the recombinant protein works in the GNE myopathy. Right now, we're just trying to see if there is a repair defect. But if there is, that would be the logical next step, right? To see if there's a protective response there, because it would be great to see this move on and potentially be a therapeutic approach that would be useful in GNE myopathy, but or something that could work alongside even a gene therapy. Because, like I said, most gene therapies can't even be envisioned at being 100% effective at curing the disease. They're like any other therapy. They have a therapeutic index, and they provide a certain amount of therapy, but there's always another way. One of the other areas we work in my laboratory is heart failure. And in heart failure, most heart failure patients aren't on one drug. They're on 3, 4, 5, some cases 7 different drugs. And the reason is there's a lot of different ways to manage the disease state. And my expectation, and I think the expectation that the field is moving towards, is that there will be multiple treatments used in neuromuscular patients regardless of what type of gene therapy it is. We're always going to have to be able to help manage the remaining symptoms using other therapeutics. And that's one potential way we could see this going forward in GNE myopathy.
Yeah, so you're a little bit prophetic because you keep touching on the next question with your last answer. So the next question we had was, would GNEM
That's because they're good questions. Everyone's listening. So, you know.
Would GNEM membrane repair be used in conjunction with gene therapy as a cure?
Yeah. So that would be a great way to think about it, right? So people call them adjuvant therapies. That's one way to think about it. But yeah, you would want to be able to use I mean, we think that in certain circumstances, you see that in some of the disease states that we've looked at here in the animals, that you can use it as a primary therapy. But in any case, one of the things that I find exciting about the work that we're doing in the lab now is the fact that you can see this working in a lot of different types of diseases, right? You know, you see, when you talk about gene therapy, that's very specifically targeted towards a particular type of disease, right? You're doing a gene replacement for one type of a gene, and that's one of the challenges we've already talked about in this series too, right? Is that if you're going to do gene therapy for GNE myopathy, that gene therapy is going to target just GNE myopathy, and Lali and others have talked about the challenges that biotech companies see in that, because it's a relatively small patient population, and getting the investment to be able to do that sort of a development for what's a relatively small patient population is a hard thing to do. And I think, you know, the NDF is working hard at that, and the researchers in the field are working hard at that, but it's very difficult. When you see the sort of things where you can apply it across a broad spectrum of different disease states. Maybe, for example, in this case, MG53, but other attempts too, like anti-inflammatories, and there's other approaches. When you see those and they can be applied across a broad spectrum of different types of neuromuscular disease, maybe they won't have the therapeutic index. Maybe they won't be as much of a cure as you see with gene therapy. But if you stack enough of those together, maybe you can have a similar response. And sometimes with these sort of rare diseases in the neuromuscular space, if you can target multiple ones at the same time, if you can increase that patient pool, you can really lead to having a situation where you can interest a lot of biotech companies a lot more efficiently, because that's a bigger patient population and they can recoup their costs a lot more efficiently. So that's one of the things that I really like about this idea of adjuvant therapies. And I can certainly see the recombinant MG53 as an adjuvant therapy for a lot of diseases, including potentially GNE myopathy as well.
Makes sense. It's so exciting. So this is our last question, and it's probably a question that you expect to hear, and that a lot of people probably have for you that you probably can't really answer. But when do you expect an IND for MG53?
Oh, well, that's a great. Yeah, you're right. That is a question that we get a lot, and something we push for. So just to define the term, so an IND is an investigational new drug application. These are something you file with the Food and Drug Administration to get access. So you're allowed to use whatever your potential therapeutic is in a patient population. So it's the gateway to be able to do a clinical trial in patients, and something everybody wants to do therapeutics development. That's one of your goals, to get that in patients and see if it works there. So that's something that we are very interested in. And as I mentioned, we're working on a next generation, which is kind of what we want to push forward into that IND process, because we don't want to take something that's suboptimal, because you don't get a lot of chances to do this sort of thing, right? Even in a career, you don't get a lot of chances to move something forward into patients. So we want to have the best molecule, and we want to move that forward as quickly as possible, but also make sure we have the right thing to treat patients, and that it'll be efficient and that we can get on the market. Those are all key factors for moving forward. So right now, I mean, I'd see at least a couple years out before we would be able to get this into an IND process. I think that's something we can move faster. I mean, we're working, as I mentioned earlier, we're working with different companies to try to move this along. But there's only so much you can do, and there's obviously funding helps to move that faster, but there are inherent difficulties in terms of moving forward as quickly as we would all like. So we're certainly pushing towards that. I could see an IND at least a couple years down the road, but something we definitely want to pursue, and we think could be of great benefit to different patient populations.
Right. Well, I think that everyone will look forward to an update from you whenever you have news to share, for sure. I think you've covered most of the questions. I think in the chat now, it's mostly just thanks so much for such an informative and wonderful presentation. I think everyone gained a lot of information from it, and will enjoy watching it again. We'll have this and all of the rest of our Symposium Speaker Series events will be housed on our video library on our website, CureHIBM. You'll be able to see this one as well very soon. Noah, I can't thank you enough for joining us today and for giving your time and energy to our patient population. It's appreciated more than I think you could possibly know, and we look forward to our continued collaboration with you and your lab.
Well, thank you. I mean, this has been a great opportunity. I really appreciate it. And I really appreciate the opportunity to both talk to the patient population and the scientists working on pursuing the cures for GNE myopathy. So thank you very much.
Thank you. Thank you. Yes, we have a wonderful group of scientists working with us. Well, we all feel really lucky to have you all invested in our mission. With that, we will let you go on to your days, and thank you all for tuning in and for your continued support. Like I said, you can watch the recordings of these episodes on CureHIBM in our video library, and hopefully we'll see you all next week. Thank you. Thanks, Noah.
Thank you.